In this video tutorial, the synthesis of imine and enamine compounds will be overviewed. This is an MF.Mer group production. In general, amines are compounds that contain a carbon-nitrogen double bond. For amines that possess a substituted carbon group, rather than a hydrogen atom attached to the nitrogen, such amines are also known as Schiff bases. A general amine structure is shown here where the R groups are typically either hydrogen atoms or substituted carbon groups, or a combination of the two. Note that both E and Z stereoisomers are possible. That is, the R double prime substituent on the nitrogen atom may either be on the same side as the R substituent, as in the structure shown here, or on the opposite side. Equilibration of the E and Z isomers can oftentimes be readily achieved. As you will see, amines are typically derived from aldehydes or ketones. Amines that are derived from aldehydes are sometimes called aldamines, while amines derived from ketones are occasionally referred to as ketamines. Similar functional groups that also feature a carbon-nitrogen double bond include oximes, hydrozones, and semicarbozones. A general synthesis of an amine is shown below. Here, an aldehyde or ketone is shown to react with a primary amine. A primary amine bears two hydrogen atom substituents and one carbon-centered substituent. This reaction is an equilibration that typically occurs under mild reaction conditions. Nevertheless, the reactions are fastest under weakly acidic conditions, typically around pH 4 to 5. Under strongly acidic conditions, the amine would be protonated and could not participate in the reaction, as we will see shortly in the reaction mechanism. The result of this transformation is the bonding of the amine and aldehyde or ketone compounds via a carbon-nitrogen double bond. Note that amine formation is reversible. The reverse process is referred to as imine hydrolysis. To minimize imine hydrolysis and maximize the yield of the amine, water is often removed from the reaction system as it is formed. A specific example of an amine synthesis involves the reaction of benzaldehyde with aniline under mildly acidic conditions. The reaction results in the formation of the amine known as N-benzylidine aniline. Water is concurrently formed as a byproduct. Shown here is a general amine hydrolysis reaction. By an acid-catalyzed mechanism, an amine will react with water. Amine hydrolysis is favored when excess water is present. The result of this reaction is the formation of the corresponding aldehyde or ketone and a primary amine. Now let's look at the multi-step reaction mechanism of amine formation. First, the electron-rich primary amine acts as a nucleophile and attacks the carbonyl carbon of the aldehyde or ketone due to the partial positive charge on the carbonyl carbon. The result is a neutral Zwitterion with both cationic and anionic character. Next, an intramolecular proton transfer is shown within the Zwitterion to result in the formation of an unstable carbonylamine. This proton transfer need not be intramolecular. Rather, the equivalent proton transfer could occur through two or more intermolecular proton transfers. In the next step, protonation of the hydroxy group of the carbonylamine converts the hydroxy group to a stable leaving group, that is, water, which is lost in the next step. Elimination then occurs and results in the formation of the carbon-nitrogen double bond and loss of water. This intermediate is known as an iminium ion, and it is the resonance stabilized carbocation. Deprotonation of the iminium ion by water results in the amine product and regenerates the hydronium ion acid catalyst. Note that each step of this overall reaction mechanism is reversible, and that the overall reverse of the steps shown here constitutes the mechanism of imine hydrolysis. Several imines play important roles in the chemistry of biological systems. For example, an imine-containing protein, known as rhodopsin, is involved in the chemistry that underlies human vision. Shown here is 11-cis-retinal. It is derived from vitamin A and features an aldehyde. Within the eye, the aldehyde of 11-cis-retinal reacts with the primary amine of the high molecular mass protein known as opsin to produce rhodopsin. Upon absorption of a photon of appropriate wavelength, the 11-cis configured alkene is isomerized to the 11-trans configured alkene. Along with this reconfiguration, a nerve impulse is sent to the brain. The nerve impulse is processed as a visual image. The general structure of an enamine is shown below. An enamine is a compound that features an alkene bonded to an amine, thus enamine. More specifically, for enamines, an amine group is bonded to one carbon that is then further doubly bonded to another carbon group. A general enamine synthesis is shown below. An enamine may be formed from the reaction of an aldehyde or ketone with a secondary amine. In this case, it is necessary for the aldehyde or ketone to have an alpha carbon with an alpha hydrogen. This reaction is fastest under weakly acidic conditions, as with the previously described amine synthesis. The result of this synthesis is the formation of a carbon-carbon double bond with a nitrogen substituent, and, as with the amine synthesis, this enamine synthesis is also a reversible process. The reverse reaction to this enamine synthesis is referred to as enamine hydrolysis. A specific example of the synthesis of an enamine is the reaction of phenylacetaldehyde with pyrrolidine under mildly acidic conditions. The reaction results in the enamine product E1-styrylpyrrolidine and water as a byproduct. Let's also work through the multi-step reaction mechanism of enamine formation. Initially, the secondary amine acts as a nucleophile and attacks the carbonyl carbon of the aldehyde or ketone. Next, an intramolecular proton transfer is shown to result in a carbonyl amine. Protonation of the hydroxy group of the carbonyl amine sets the stage for the elimination of water in the following step. Lastly, water is shown to act as a base and remove a proton from the alpha carbon, thus providing the enamine product while regenerating the acid catalyst. In summary, both of these common imine and enamine syntheses involve the reaction of an aldehyde or ketone with an amine. Primary amines are required for these imine syntheses, while secondary amines are typically used to form enamines. Both reactions are optimal under mildly acidic conditions. If the conditions are too acidic, the reaction rate will decrease. Importantly, both reactions are reversible in the presence of water. All of these features, including the generally mild reaction conditions, thus renders imine and enamine chemistry prominent in biochemical reaction sequences as well as in industrial applications.